Okay, well, here we go. So what I want to do is, if you've been following me for a while, which is very unlikely because I only have 5 subscribers, but if you've been following me, you probably noticed that I like to go over review papers, and the reason why I like to do that is the authors of these review papers are experts, pretty much experts in their field, and they've spent a lot of time poring over the literature to know as much about a particular subject as possible, so they have a pretty comprehensive understanding of the subjects that they write about. And what I like about the reviews is that these experts will put in as much information as they believe is necessary to have a comprehensive understanding of that subject. Okay, so because of this, reviews are really an excellent way to learn a lot about a subject in one of the most concise formats. And yeah, really, if you can understand reviews, you can pretty much learn a lot about, or how do I phrase that? You can start to approach the understanding of a doctorate in that field, which is, kind of humbling, but at the same time, it is very, I forgot who it was that said it, but we basically get to stand on the shoulders of giants by reading these reviews, because we are able to look at all the research that had been done and analyze it in from a holistic point of view. So, that's one of the reasons why I like to go over review papers, and that's also one of the reasons why I believe Wikipedia is doing as well as they are, because to a degree, every Wikipedia article functions somewhat like a review article on that particular subject. So this is one of the reasons why I believe Wikipedia is as big and as broad as it is today. And regardless, so what I want to do is go over this review, and I've actually, I really can't verify the legitimacy of, or I should say I can't verify the impact factor of this particular journal, the Asian Journal of Pharmaceutical and Clinical Research, but I will go through this one, for one, because it's in the Creative Commons. It is the most up-to-date review paper that I found on this subject, which the subject is the major histocompatibility complex. And remember, this is not my field, so if they have made mistakes in this, then I am absolved of, gosh, what would the word be? If there are mistakes in this paper, I won't hold myself responsible for being able to refute the claims that are being made in this paper. Okay, so one of the reasons why I wanted to go over the major histocompatibility complex is because it plays an extremely important role in immune function. And so if we can understand this, we can start to understand a little bit better immunity and autoimmunity, and how disruption of that entire process can screw people up pathologically. Okay, so with that out of the way, I am going to jump into it. So the major histocompatibility complex, a review by these people. So abstract, one of the important components of the immune system, the major histocompatibility complex molecule, allows T lymphocytes, which is a type of white blood cell, to detect cells such as macrophages, B lymphocytes, and dendritic cells that ingest infectious microorganisms, so they take up the microorganisms, or the cells infected with microorganisms. So not only can, how do I phrase that? The major histocompatibility complex is allowing the T lymphocytes to detect cells such as macrophages, B lymphocytes, and dendritic cells, and these cells are ingesting microorganisms or they are ingesting human cells that are infected. So if everything's working correctly, if a cell gets infected with a virus, or if there is some form of parasite that gets into the cell, then hopefully they get eaten by macrophages, B lymphocytes, or dendritic cells, which are also a part of the immune system, and then they will present the pieces of that pathogen on their surface in order to basically activate these T lymphocytes. Okay. Anyways, on being engulfed, a microorganism on being engulfed by a microorganism. No, sorry. Like I said, if there are mistakes, I don't. If there are mistakes, I can't really control for that. So on being engulfed, on engulfing a microorganism, I believe is what they tried to say. Macrophage partially digests it and displays the peptide fragments of the microbe on its surface. Okay, so this is something that's important to consider, is that a pathogen gets consumed by the macrophages, and then the macrophages will partially digest these pathogens, and then it will present these pathogens on its surface, and I should say it'll present the peptides of the pathogen. So take up the pathogen, break it up partially, and then present them on the surface, and then the presentation of those peptides is something that can teach the immune system what it needs to attack. Okay? And then bound to the major histocompatibility complex molecules, and the T lymphocyte recognizes the foreign fragment attached to the major histocompatibility complex molecule and then binds to it. And then this binding of the T lymphocyte to the MHC leads to stimulation of an immune response. The major histocompatibility complex molecule presents peptides from its own cell. These are called self-peptides in healthy self cells, to which T cells do not normally react. So the major histocompatibility complex will present peptides from itself, but it's done in a way to convince T cells not to attack these. So the major histocompatibility complex is serving two functions. With one function, it will present the peptides of your own body and then convince T cells not to attack those peptides. And then the other function is to present peptides of a pathogen and convince the T cells to attack that pathogen. Okay, so this is one set of molecules that is fulfilling dual roles. And by proxy, it is possible that this signaling can get screwed up, and that's how you could theoretically get some autoimmune diseases to take place. Okay, but let's continue. So, introduction. The major histocompatibility complexes are a class of molecules that influence the capability of an organism to accept or reject transplanted tissues. In contrast to the B cell receptors and antibodies, which recognize the antigen single-handedly, the T cell receptors only recognize the antigen processed and presented by Mhc molecules. So B cells and antibodies can attack antigens by themselves, in of themselves, but the T cell receptor can only recognize the antigen when it's been processed by the Mhc molecules, according to these people. The function of the Mhc molecule includes its binding to a peptide fragment derived from some pathogenic microorganism. So the microorganism has a bunch of proteins in it, and so you break up those proteins into smaller peptides. And then those smaller peptides, if the signaling is working the way it should, those small peptides can bind to this major histocompatibility complex, and then it presents that it will present that peptide to the surface. And by presenting it to the surface, then it can interact with T cells. Okay, so displaying of these antigens happens on the cell surface for its recognition by some suitable T cells. These cause deleterious effects on the pathogen, and on the pathogens, virus-infected cells are killed, and macrophages are also induced for killing bacteria to other pathogens living in their intracellular vesicles. Okay. So, parallel to these happenings, B cells are activated to generate antibodies that remove or neutralize extracellular pathogens. In course of all these events, or during all these events, there are chances in favor of any pathogen that has mutated in such a way so as to escape from presentation by major histocompatibility complex molecules. So what they're saying here is it may be a selective pressure for organisms to avoid this MHC process, because if the peptides end up going into this MHC process, it ends up screwing with the immune system's response. Anyways, Mhc molecules decide upon the response of an individual to antigenic infection. Mhc molecules will decide which response an individual will take to an antigenic infection. Thus, its role is implicated in the susceptibility to the disease, as well as in autoimmune disease. So if this process gets screwed up, an antigen might be presented that ends up convincing the T cells to attack when really you did not want the T cells to attack. So that's something, and if that peptide looks similar or is one of your own peptides, that can cause the T cells to attack something that it really shouldn't be. Anyways, human leukocyte antigen is the expanded version of HLA, and this HLA is similar to the major histocompatibility complex in structure and function, and is a group of antigens or proteins that are found at the surface of cells and in the genetic makeup of DNA. Maybe this wasn't a good review to use. It's a little too many verbiage problems that make it a little difficult to keep up. But one second. So HLA is a group of antigens or proteins that are found at the surface of cells and in the genetic makeup of DNA. A T cell recognizes peptide antigen, which is attached by a specific kind of allelic variant of a Mhc molecule. It will not recognize the same peptide bound to other Mhc molecules, and this quality of T cells is called Mhc restriction. So let's look this up just to get a little bit more education. So Mhc restriction, antigen recognition, or Mhc restriction refers to the fact that a T cell can interact with a self-major histocompatibility complex molecule and a foreign peptide bound to it, but will only respond to the antigen when it is bound to a particular Mhc molecule. So, a little background. When a foreign protein enters a cell, they're broken into smaller pieces called peptides, and these peptides are known as antigens, or also epitopes sometimes, and can derive from pathogens such as viruses or intracellular bacteria. Anyways, foreign peptides are brought to the surface of the cell and presented to T cells by proteins called the major histocompatibility complex. During T cell development, T cells go through a selection process in the thymus to ensure that the T cell receptors will not recognize MHC molecules presenting self-antigen. So in the thymus, where you have the maturation process of a lot of these T cells, if the T cells must go through a validation process by which they will, if they can recognize your own peptides, I don't know if they get killed or if they get suppressed, but either way, that is the way at which the body filters out autoimmune T cells. Okay, or rather, it has a moderate affinity. So yeah, there could be some affinity, but it's not a good affinity. Anyways, high affinity means it will be autoreactive, or it will attack your own body. But no affinity means it will not bind strongly enough to the major histocompatibility complex. The selection process results in developed T cells with specific TCRs that might only respond to certain MHC molecules, but not others. Okay. The fact that the TCR, and what does TCR mean again? That's the T cell receptors. The fact that the T cell receptors will recognize only some major histocompatibility molecules, but not others, contributes to the Mhc restriction. Okay. So let's go back. They are the biological tools meant to identify and prevent a foreign protein or cell in entering or its spreading in an organism's body. Being one of the important components of the immune system, the proteins recognize between self and non-self. So this is important, recognizing between what is your own peptides and what is not your own peptides. So the major histocompatibility complex molecules are often present in vertebrates, while HLA is only found in humans, and it can be better explained as the human body's version of the major histocompatibility complex. Anyways, local cells carrying viruses and other harmful microorganisms are often identified with the help of these major histocompatibility complexes, or HLA molecules, and are attacked. Many times, these antigenic molecules are involved when an organ transplant is planned for an organism or a human being. So these antigen-presenting molecules are of great importance whenever we do transplants, because if you have a transplant and the organ is presenting a lot of antigens on the surface, if those antigens are dissimilar enough from your body, that can cause a lot of problems for the recipient of that transplant. Anyway, some tests are carried out to determine the compatibility between an organ and a recipient's body, and near perfect or perfect matches are desirable in these situations to lessen the risk and also the chances of the recipient's body in organ rejection. So if your immune system starts attacking your transplanted organ, it's probably going to fail. It's probably going to end up dying within you. So it's important that we test to see if it is as compatible as possible. And on top of that, we usually get immunosuppressants to help the body handle that foreign organ as well. But yeah, you'd like to start with the most compatibility as possible. Anyways, therefore, scope of both major histocompatibility complex and HLA, which is the human's version, in organ transplants lies in making our body and its defense stronger. Besides its important role in immunity, HLA is also used in determination of the parentage of a child by comparing antigens from the child, father, and mother. Interesting. So in paternity testing, earlier studies have shown that MHCs are derived from a cluster of genes and play an important role in both cell-mediated and humoral immunity. These clustered genes lie on long continuous stretches of DNA on the chromosome 6 among the human beings, and it is on chromosome 17 in mice. Okay? In humans, they are also known by the name HLA complex, and major histocompatibility gene encodes for 3 major and important classes of molecules, and these classes are 1, 2, and 3. Class 1 major histocompatibility genes encode a particular glycoprotein on every nucleated cell of the body, and major played by this class of molecule is the presentation. Don't think that was the right word, but by this class of molecule is the presentation of peptide antigens to cytotoxic T cells, while class 2 major histocompatibility genes encode for the glycoproteins, which are expressed only on the surface of antigen-presenting cells. So MHC class 2 is specific for antigen-presenting cells, or APCs. So these are, I believe they're all immune cells. So that is type 2. And type 1 is on every cell of the body. So type 2 is for the immune cells, but type 1 is on every cell of the body. Okay, so these we'll go into more detail later. And these antigen-presenting cells can be macrophages, dendritic cells, and B cells. Here, antigenic peptides, after being processed, are presented to TH cells. Let me look this up just to double check what. Oh, the T helper cells, okay. Anyways, class 3 major histocompatibility complex gene encodes various proteins that play important immunological roles, and also the components of complement system, including also the components that complement systems, including proteins involved in inflammation, are being mediated by these. Whew. Anyways, the class 1 and 2 molecules have important roles in antigen processing and presentation, while class 3 regions, which is flanked by the class 1 and 2 regions in the gene, is responsible for the expression of molecules concerned with immune function. Yeah. There are a number of complement factors, including C2, C4, factor B, and several inflammatory cytokines, including tumor necrosis factor, which is expressed by the mediation of the class 3 major histocompatibility complexes. So, a little difficult to follow, but I think we got it. Anyways, MHC class 1 molecules. So structurally, the MHC class 1 molecules are heterodimers. So there are 2 pieces that are different that come together to form the final molecule. It consists of 2 polypeptide chains, alpha and beta-2 microglobulin, or B2M, as heavy and light chains. So the heavy chain is the bigger chain, and the light chain is the smaller one. So the alpha is the heavy chain, beta-2 the light chain, respectively. Anyways, the 2 chains are linked non-covalently, so there's no covalent bonds between this heterodimer. The 2 chains are linked non-covalently through interactions of beta-2 microglobulin and the alpha-3 domain. So this is the 3rd domain of the alpha peptide chain. The alpha chain is polymorphic in character and encoded by an HLA gene, while the B2M subunit is not polymorphic and is encoded by the B2M gene. So polymorphic means there's a lot of different forms of it. So this A chain, there's a lot of variability in it. But the B chain the beta chain is consistent. It doesn't, it's not trying to be variable. It could theoretically be variable by a mutation here and there, but it's not attempting to be variable. Anyways, the alpha-3 domain spans over the plasma membrane and interacts with the CD8 co-receptor of T cells. So the CD8 is one of the receptors on T cells. Okay, just keep that in mind. And this interaction holds the major histocompatibility complex 1 molecule in place. So there is a portion on the MHC1 molecule that is on the alpha chain, specifically the third domain, the alpha-3 domain, and then this alpha-3 domain will go through the plasma membrane and it will interact with the T cells CD8. And that interaction will hold the major histocompatibility complex of whatever cell it's on and the T cell, okay? And it will just hold them together, at least momentarily. The other kind of receptor, called the T cell receptor, lies on the surface of the cytotoxic T cells, and it binds its alpha-1 and alpha-2 heterodimer ligand and checks the coupled peptide for antigenicity. So here, the T cell binds its alpha-1 and alpha-2 heterodimer ligand and checks the peptide being what is presented on the major histocompatibility complex, and it will check to see if that is a good or bad peptide, okay? Or I should say, it will check to see if it latches on or not. That would be a better way to describe it. Anyways, the alpha-1 and alpha-2 domains fold in a manner to make up a groove for peptides. So this alpha-1 and alpha-2 domains on the alpha chain of MHC is variable. Keep in mind, the alpha chain is variable, and they have grooves for which a peptide can fit in nicely. Okay? So, and these grooves can be either of your own body. They can fit peptides from either your own body or from a pathogen of some sort to bind. So the major histocompatibility complex class 1 molecule binds peptides that are about 8 to 10 amino acids in length. The peptide translocation from the cytosol into the lumen of the endoplasmic reticulum, and the lumen is the area of a cell that is within the endoplasmic reticulum, is accomplished by the transporter associated with antigen processing, so TAP, transporter associated with antigen processing, which is a member of the ABC transporter family and is a heterodimeric multimembrane spanning polypeptide consisting of TAP1 and TAP2. So remember, it is a well, it's a dimer of some sort. The two subunits form of TAP are peptide binding site and two ATP binding sites that face the cytosol. Okay? The two subunits form a TAP. Okay, I think there's a wording issue, but the two subunits will remember this protein spans multiple membranes, right? And these two subunits of TAP will present a peptide binding site and two ATP, which is an energy currency of the cell, and two ATP binding sites, and these face the cytosol. So not the lumen, they face the cytosol. The antigenic or non-antigenic peptides are now bonded with this transporter associated with antigen processing, or TAP protein, on the cytoplasmic side. And then these are translocated by consuming ATPs, so basically the ATPs are used as energy currency to translocate the peptide into the lumen of the ER. And there, the major histocompatibility complex class 1 molecule is in turn loaded with the peptides in the lumen. So this happens in the endoplasmic reticulum, not in the cytoplasm, or the cytosol, I should say. This complex phenomenon of peptide loading involves several other molecules that form a large multimeric complex consisting of, okay, well, it doesn't matter too much, but tapicin, calreticulin, calnexin, and ERP57. Calnexin acts to stabilize the class 1 major histocompatibility complex alpha chains before the beta-2M binding. So remember, the class 1 MHCs have an alpha chain, a large variable alpha chain, and a beta-2M chain. The beta-2M chain is the same for all of them, but the alpha chains are unique. And before the beta-2M chain is bound to the alpha chain needs to be stabilized, and calnexin within the lumen of the endoplasmic reticulum is fulfilling that function. Anyways, following complete assembly of the Mhc molecule, calnexin dissociates, and then the major histocompatibility molecule, which is lacking a bound peptide, is inherently unstable and requires the binding of the chaperones calreticulin and ERP57. The protein tapicin binds to the Mhc molecule to link to the TAP proteins to facilitate enhanced peptide loading and co-localization. So let me read this. The protein tapicin binds the Mhc molecule to link it to the TAP proteins, and this is to facilitate enhanced peptide loading and co-localization. So tapicin is just trying to get the alpha chain close to the TAP proteins, and remember the TAP proteins have the antigen in them, so the co-localization, or getting those things together, is important. So reaction rates, so in chemistry, a lot of reaction rates are associated with concentration, right? The more concentrated your chemicals are, the faster the reaction will take place. While in biochemistry, frequently to make reactions go faster, we will bring the chemicals together, and by bringing the chemicals together, it's as if the concentration of those chemicals is higher in that small area. And so by co-localizing or bringing these two proteins together, there is a facilitation of that interaction to take place. Anyways, on being loaded with the peptide, the major histocompatibility complex class 1 molecule, the complex dissociates and it leaves the endoplasmic reticulum through the secretory pathway. So secretory pathway is the pathway by which proteins leave the endoplasmic reticulum to be secreted out, and it happens in a process of forming vesicles that move further and further away from the endoplasmic, or further away from the nucleus, more towards the cellular membrane, where it eventually will be either released or become a part of the membrane. Anyways, the transport of the Mhc class 1 molecules through the secretory pathway involves several post-translational modifications of the Mhc molecule. So remember, post-translational modifications. Remember, we go from DNA, the genetic material of the cell. The DNA will get transcribed into RNA, messenger RNA specifically. You can think of it as copying the notes of the DNA, and then those notes, those messenger RNAs, will get translated into the proteins, right? So that's translation, going from the mRNA into production of proteins, which are the machinery of the cell. And then sometimes after that translation process, sometimes the protein is not in its final form. It's still not in its final form, right? So to get the proteins in their final form, there is often a lot of post-translational modifications. And these can include glycosylation or different types of phosphorylation or proteolytic cleavage. There's a laundry list of things that can happen to the proteins after they've been produced. So these are called post-translational modifications. And in this case, it's of the major histocompatibility complex molecule. And these involve changes to the N-glycan regions. N-glycan meaning nitrogens that are bound to sugar molecules, N-glycan, regions of the protein, followed by extensive changes to the N-glycans in the Golgi apparatus. So even after you add these sugars or these carbohydrates or oligosaccharides to these nitrogen groups on the protein, they still need to go through more processing as it leaves the Golgi apparatus. Anyways, the N-glycans form is a completely mature form before they reach the cell surface. Figure 1 shows the processing and presentation of the antigen by Mhc Class 1 molecule. So this is Figure 1, okay? So this is good. So here, this is the mRNA of the alpha chain of the Mhc molecule. So this particular region is very error prone. There's a high rate of error in translation. So this is one of the ways that we can create a lot of variability in the proteins that are being produced is by this high error rate, right? So what happens is you have a lot of error in your alpha chain production. And then here, it goes through ubiquitination, which then tags it to be proteolytically cleaved in some way. So gets tagged by ubiquitin, and then gets cleaved by proteasomes, and then you get these peptide epitopes, these little peptides. And then these peptides get shuttled in by the TAPs or the TAPs, and this tapicin will bring the TAP and oops, I misspoke. Yeah, I misspoke. So here, I'm interested that this is happening at, okay, 38 minutes. So here we have a peptide epitope, and there's a high error rate in translation. And then this is producing a protein peptide, right, by the ribosome, by translating this mRNA. And then ubiquitin comes along, or it gets ubiquitinated, and then this signals to the cell that it needs to proteolytically cleave this epitope, or this area of the amino acid chain. And that is done through the proteasomes. And then here you have your peptide, and it gets, or I should say, you have your epitope, and then this epitope gets cleaved by peptidases into smaller peptides. These peptides will interact with your TAP, your TAPs, and then this tapicin will bring together your major histocompatibility complex and your TAPs with the peptides close together, which is what allows this peptide to be put on the major histocompatibility complex. and this is the peptide loading complex. Okay? So that is the production of the peptides, but here is the production of the alpha chain of the Mhc Class 1 molecule. So here you have your synthesis of the Mhc Class 1 molecules. This is another ribosome, and it gets shuttled, the peptide gets shuttled in to the endoplasmic reticulum because there is a signal, there's an amino acid signal that would signal to the ribosome or to something in the cell to bring it into the endoplasmic reticulum. I forgot exactly what amino acid sequence that was, but it gets put in, and then through calreticulin and ERP57, this brings this whole what would you call it, complex together to have your Mhc Class 1 with the peptide that interacts with it nestled in nicely. And then, once this is done, it ends up getting secreted, and then it gets secreted into these vesicles, such as the Golgi complex, where it will then undergo more post-translational modification, and then it gets put out onto the plasma membrane or the cellular membrane. Okay, and now it's out and about, ready to be interacting with T cells. Okay. Anyways, the major histocompatibility complex class 2 molecules. Excuse me. So Mhc class 2 consists of 2 polypeptide chains, alpha and beta, each having 2 domains, alpha 1, alpha 2, and beta 1 and beta 2. Each chain has a transmembrane domain, alpha 2 and beta 2, respectively, for attaching to the major histocompatibility complex class 2 molecule to the cellular membrane. So the alpha 2 chain, or the alpha 2 subunit, the alpha 2 domain of the alpha subunit and the beta 2 domain of the beta subunit are the portions of those two molecules that interact with the cellular membrane. Okay, that's what keeps it anchored to the cellular membrane. The peptide binding groove consists of the heterodimer of the alpha 1 and the beta 1. So this is in contrast with major histocompatibility complex 1, where the groove was formed within the alpha chain in of itself, but for the class 2, the groove comes from the interaction of the alpha 1 domain and the beta 1 domain. So it's coming from both peptides. Anyways, Mhc class 2 molecules are normally occur only on professional, that's a word, antigen-presenting cells. So the antigen-presenting cells are the immune cells who have the explicit function to present these antigens in order to modulate immune activity. Okay, such as and these APCs, these antigen-presenting cells, are macrophages, B cells, but it is especially the dendritic cells. So this is the main function, one of the main functions that only the dendritic cells are really good at, because the dendritic cells are known to be presenting these antigens either in a pro-inflammatory or pro-attacky way or anti-attacky way. But I wonder if they'll go through that more in this paper. Anyways, although they can be conditionally expressed by all cell types, so all cell types can theoretically express these Mhc class 2 proteins, well, conditionally, it could happen under certain circumstances. But an antigen-presenting cell takes up an antigen, performs antigen processing, and returns a molecular fraction of it, termed as the epitope, to the APC surface. And this is coupled with an Mhc class 2 molecule. The process is known as antigen presentation. And yeah, they have a figure down there. So this is, you can see it in figure 4. So let's go to figure 4 just to, yeah. So here's figure 4. Here's your antigen presenting cell. And here you have the class 2 histocompatibility molecule with the peptide in it. And this particular one will have a little binding site for the CD4 receptor on the T cells to interact with. And then the T cell receptors will also have grooves on them to hopefully bind this peptide. Anyways, so, and this is the structure of it. Both the alpha and the beta chains will create this groove of the histocompatibility complex. Anyways, going back, in this unique presentation, the epitopes are recognized by T helper cells, the CD4 receptors, as well as the T cell receptors are present on the surfaces of helper T cells. When a CD4 molecule of a naive helper T cell, so this is the naive helper T cell, or the young immature T cells, when the CD4 molecule of a naive helper T cell links to an APC's major histocompatibility complex 2 molecule, its T cell receptor can meet the epitope coupled within the Mhc Class 2. The phenomenon, similar to the coupling of the cytotoxic T cells with cytotoxic cells. Okay. This event primes the naive helper T cell. The varied combination of cytokines secreted by antigen-presenting cells in the microenvironment causes the naive helper T cell to polarize into either a memory T cell or an effector T helper cell. So, what is a memory T helper cell? Memory T cells are a subset of the T lymphocytes, and that might have some of the same functions as the memory B cells. Their lineage is unclear. Antigen-specific memory T cells against viruses or other microbial molecules can be found in both the TCM and the TEM subsets. Okay, so it's getting a little too deep. I'm going to backtrack a little bit. And then the effector T cells the cytotoxic are the CD8 presenting, and the helper are the CD4 presenting. Okay, well, yeah, so all this stuff is still a little unknown, but we'll keep on going. So cytokines are secreted by the antigen-presenting cells. And so these cytokines can tell the cells whether or not they should be attacking or not. How do I phrase this? There's a lot of hormones and a lot of cellular signals that these antigen-presenting cells secrete, which can help tell the immune system what course of action they need to take, given those particular, given the particular peptides that they are showing. Okay, so that is the main takeaway. So I don't think it's here, but say an immature dendritic cell, which is an antigen-presenting cell, an immature dendritic cell will frequently express antigens. But if the environment is beneficial at that time, a lot of those antigens might be considered safe for the body. They're safe antigens. But in the context of a pro-inflammatory environment, which could happen through a lot of cell necrosis or a lot of apoptosis or a dangerous pathogen coming in, that can help turn the immature dendritic cells into mature dendritic cells, and then that would cause the differential expressions of cell receptors. And that different expression of cell receptors can then warn T cells, oh yeah, these antigens are associated with bad things, and therefore you should attack these antigens. So when a naive T cell interacts with a dendritic cell that is saying attack these, it'll go from a naive T cell to a mature T cell. Anyways, I think we should keep on going. Anyways, major histocompatibility class 2 thus mediates immunization to antigen-presenting cells. They also differentiate the T cells into memory and effector cells, so that in the future, if the same antigen invades the organism, the memory cells get activated and trigger the immune response. So the memory cells will get activated when they see this antigen again. The polarization during primary exposure to an antigen is key to determining a number of chronic diseases. This is including inflammatory bowel diseases and asthma by inducing the immune response that is the memory T helper cells. That memory T helper cells coordinate when their memory recall is triggered upon secondary exposure to similar antigens. So remember, this doesn't actually have to be pathogens, it just means antigens. So food allergies can cause this, or pollen can cause this. Bee stings can cause this if you're allergic to that. So really, you could be allergic to anything. Just by pure chance, somehow some wires got crossed in this antigen presenting process, which results in allergies or allergic reactions, hyperinflammatory reactions that would not normally exist. Anyways, in the phase of processing and presentation of the antigen, the phagocytes, such as the macrophages and immature dendritic cells, take up antigenic entities by phagocytosis and phagosomes. B cells exhibit the more general endocytosis into the endosome. Okay, so immature dendritic cells take up antigenic entities by phagocytosis into phagosomes, but the B cells exhibit the more general endocytosis into the endosome. Okay, the phagosomes, which are of the dendritic cells, or endosomes, which are of the B cells, fuse with lysosomes, which is like the stomach of the cell. And its acidic environment, or its acidic enzymes, will cleave the uptaken proteins into many different peptides. And out of these, if a particular peptide exhibits immunodominance and is loaded into the Mhc Class 2 molecules, out of these, a particular peptide exhibits immunodominance. And I should say, out of these, there could be a peptide which exhibits immunodominance, and it is loaded into the Mhc Class 2 molecules. So these antigen-presenting cells express both kinds of major histocompatibility complex 1 and 2 molecules. However, in the cell, there is a highly advanced system that exists to prevent Class 2 molecules from binding the same set of antigenic peptides as the Class 1 molecules. So there is and I would assume this would be something that would help prevent autoantigenicity. So there is a mechanism in place to all the things that are on the Mhc Class 1 proteins. There seems to be something that prevents those same peptides from interacting with Mhc Class 2 proteins. So in course of synthesis of Mhc Class 2 in the endoplasmic reticulum, 3 pairs of Class 2 alpha-beta chains associated with the pre-assembled protein in a associated with a trimeric protein called invariant chain, and this invariant chain, which interacts with peptide binding cleft of the Mhc class 2 molecules, and it blocks the attachment of any endogenous peptides to the cleft. So there is a trimeric protein which is called, let me see. In the course of synthesis of Mhc class 2 in the endoplasmic reticulum, 3 pairs of class 2 alpha beta chains associated with the preassembled protein, which is trimeric, called invariant chain, and this interaction happens with the peptide binding cleft of the Mhc class 2 molecules, and this blocks the attachment of any of the endogenous peptides to the cleft. Okay. The invariant chain complex with the class 2 molecule is now transported from the RER, RER, what was that? The rough endoplasmic reticulum through Golgi complex to the endosome lysosome assembly. And then during this journey from the rough endoplasmic reticulum to the endosome lysosome assembly, the invariant chain gets degraded under the influence of proteolytic enzymes, which are, it's probably more sensitive than the other chains. And a small fragment clip, which is called clip, which is the class 2 associated invariant chain peptide, will remain attached to the class 2 cleft. And then this clip, for exchange of clip with this clip is then exchanged with the peptide that has been taken up into the endosome and lysosome. So there is requirement for non-classical class 2 molecules, which is called the HLA-DM. There is requirement of non-classical. Okay, it is a heterodimer but not polymorphic and is not expressed at the cell membrane. HLA-DM, and remember, HLA is the human version, basically the human version of the Mhc class 2, or MHCs. HLA-DM is found predominantly in the endosomal compartment. The reaction between HLA-DM and class 2 CLIP complex facilitates the exchange of the CLIP for a different peptide. Okay, so now the Mhc class molecule is uploaded with the antigenic peptide and is trafficked to and externalized on the cell surface. So this is how the Mhc class 1 can uptake the endogenous peptides more easily, because the Mhc class 1 does not undergo this complex and with the invariant subunit, or this what is it called again? The invariant chain. And by proxy, it doesn't interact with CLIP, which is preventing the peptide from interacting with the Mhc class 2. So this is how the Mhc class 2 will go all the way into the lysosome and endosomes without the peptides. Okay? So for instance, here is with the class type 1. Here you can see peptides are getting loaded in to the Mhc class 1, and this is happening in the endoplasmic reticulum, right? But our Mhc class 2 is also being synthesized in the rough endoplasmic reticulum. But we don't want it to be loaded yet. We don't want it to be loaded at this stage. We want it to be loaded when it's in when it's more in the lysosome endosome complexes with all of the endocytosed or all of the consumed peptides. So this is how we protect the Mhc class 2 molecules from getting normal human peptides inside of them. Okay, so this is how when a peptide is produced from outside of the, or how to phrase it, from a protein that was from outside of the cell, Keep in mind, sometimes your own cells, your own immune cells, will eat your own somatic cells. It'll eat the cells of your own body. So there is a possibility that some peptides from your own body could still interact with the MHCs. But we hope that that doesn't happen. Yeah. And then also, I need to mention that if, say, a virus has peptides that look very similar to human peptides, then that can present a problem with immunity, because then the virus proteins are consumed, broken down into peptides, and if those peptides look similar to human peptides, either it could be difficult for them to be presented, or how to phrase that, it could be difficult to generate any immunity at all, or if you do end up generating immunity against that amino acid sequence or those peptides, then those same, you could basically trigger the immune system to attack your own body, which would be no bueno. Anyways, the Mhc Monomers, moving fast, the tailor-made various oligomeric forms of the major histocompatibility complex molecules have been designed to identify and isolate T cells with high affinity to specific antigens amid a large group of unrelated T cells. So what happens is there are a lot of different T cells. And it just so happens when a T cell, a mutant T cell, ends up having a portion that binds very well to these Mhc molecules, this is how that T cell gets activated. Anyways, the high affinity to specific antigens amid a large group of unrelated T cells are known as the Mhc Monomers. So the T cell receptors have low affinity for their major histocompatibility complex counterparts. So initially, it was problematic to label T cells effectively using single Mhc T cell interactions. But in 1996, John Altman used a complex of multiple Mhc molecules to form a more stable bond between corresponding T cells, and it gave birth to the Mhc Monomers. So these multimeric molecules will have a range in size from dimers to octamers. So currently, few companies are using even higher quantities of MHCs per multimer to increase their specificity. These molecules are used to display class 1 MHCs, class 2 MHCs, or non-classical molecules such as CD1D from species such as monkeys, mice, and humans. Interesting. That's 27. Let me look at that real quick. So 27. Parallel detection of antigen-specific T-cell responses by multi-dimensional encoding of Mhc monomers. I might look at that later. The most commonly used Mhc monomers are tetramers, which are typically produced by biotinylating soluble Mhc monomers. Soluble Mhc monomers are typically produced recombinantly in eukaryote or bacterial cells, and then they bind to a backbone, such as streptavidin or avidin, creating a tetravalent structure. So, this really shifted gears very abruptly. So, it sounds like if you want to say, activate some T cells, bind them well, it's difficult to do it when it's just one Mhc complex. Well, okay. I'll keep on going. Let's see. So here they're trying to synthesize groups or multimeric structures with a lot of these major histocompatibility complexes. Anyway, soluble Mhc monomers are typically produced recombinantly in eukaryotic or bacterial cells, and they then bind to a backbone such as streptavidin or avidin, creating a tetravalent structure. These backbones are conjugated with fluorochromes to isolate bound T cells through flow cytometry techniques. Okay, so this is a way. This is a method. So these are soluble major histocompatibility complex monomers, which are soluble, so they're not stuck to the cellular membrane like you see over here. What ends up happening is that they have genetically engineered them to be soluble and to be secreted. And there's a way for, if you want to find a specific T cell with a specific antigen that it attacks, you can create these Mhc molecules, multimeric structures, and you can create them in a way that has a fluorescent dye attached to the structure. And when you do this, you can mix the your multimeric major histocompatibility complex what would you call it? Mass. I'll call it a mass complex. And then when it comes into contact with a T cell that has a receptor that can bind to that major histocompatibility complex, with that antigen presentation, they can go through a process called flow cytometry. And this is when cells are flowing one by one through a very small tube. And what happens is they get hit with some type of light which can cause fluorescence. And if the T cell has bound the Mhc multimer, the Mhc complex, and if this Mhc complex has a fluorescent dye on it, then what will happen is that dye will glow. And that glow will trigger, is basically a signal, and it will trigger the flow cytometry machine to record that signal. And oftentimes you can actually separate cells individually based on what is, I should say, what signals they are producing. So that this is a way to separate cells from each other. Anyways, Mhc, and you could argue that this could be good if you wanted to isolate immune cells that are very effective at binding a particular antigen. So if you wanted to, say, culture them so that you can use them to produce antibodies, or use them and inject those antibodies into someone, or if you wanted to, say, take someone's blood sample and basically get out all of the immune cells that will bind to a particular pathogen, and then you can isolate them and culture them in some way. I mean, this is starting to get out of my field quite a bit, so I don't know all the techniques that are being used right now in the medical field, but that's something that could possibly be done. Anyways, Mhc multimers are of use in antigen-specific T cell detection and isolation, and their ability gives rise to several clinical applications. So the Mhc multimers have broadened the strategy for out-of-life or ex vivo selection and proliferation of T cells specific to viral or tumor-related antigens. Okay, I was right. So this would be a way to isolate T cells that can attack a very specific antigen. And then after you isolate them, you can proliferate them. You can find ways to make them multiply a lot. And that would be useful for, say, a viral infection or a tumor-related antigen. So if someone has a cancer and you can, if someone has cancer, you can probably take out some of that cancerous tissue, and then you can find which antigens are being presented on the cancer that are different from the human normally. Anyways, these T cells can then be reintroduced to augment the immune system or make the immune system stronger for that particular against that particular antigen. Besides its vast scope in T cell selection and proliferation, the major histocompatibility multimers can also be used to eliminate graft-originating T cells on transplanted organs, ex vivo and in vivo. So this, theoretically, this could be a way that you could, that seems really difficult, but you could remove the T cells. You wouldn't do this with flow cytometry. How would you do that, though? So maybe you can put in these MHC multimers into the body and somehow attach something that can kill the cells that interacts with it? Is that... Mhc multimers can also be used to eliminate graft-originating T cells. So graft-originating T cells would be T cells that are produced as a result from a tissue graft or an organ transplant, right? So someone gets an organ transplant and they start producing T cells that are attacking this organ. That's not good. Or this graft, that would, especially if it was tissue that was there to help the person survive, it's not good if that happens. So there might be a way to use these Mhc multimers to interact with those T cells and then kill those T cells. So these Mhc multimers may also be used to eliminate harmful or unwanted T cells, such as those that target self cells. So if you have an autoimmune disease and you can make these MHC complexes that have the epitopes of whatever the target of those antibodies are, and if you can find a way to kill the T cells that way, that would be beneficial. We can do desirable modifications and also we can do desirable modifications in cancer, immunotherapy, and vaccine development by involving the Mhc multimers. Okay, so one of the things that I didn't quite like was that it went directly from MHC2 to a clinical application of the Mhc multimer, so it was hard to understand the change in pace right there. Anyways, conclusion. And yeah, it's only an hour 13, so we're doing pretty well. So the two major classes of Mhc protein molecules are Class 1 and Class 2. Class 1 Mhc molecules span the membrane of almost every nucleated cell in an organism. On the other hand, Class 2 molecules are concerned with cells of the immune system known as macrophages, dendritic cells, and lymphocytes. In human beings, these molecules are encoded by several genes which are clustered in the same region on chromosome 6, and each gene has an unusually large number of alleles, which are actually alternate forms of a gene producing alternative forms. So this area of the genome is highly mutatable. Hence, rarely any two individuals have the same type of sets of Mhc molecules. So this can be a problem for like organ transplants and such, which are jointly known as tissue type. The Mhc Class 3 genes are so diverse that they encode for other proteins such as complement proteins, cytokines, and enzymes. Besides the multimer, Mhc molecules have a diverse scope in the field of clinical medicine and research, and they can also be used to remove or arrest graft-originating T cells due to an organ transplant, and this can be done out of the life or in the life, so ex vivo or in vivo. These Mhc multimers may also be used to eliminate harmful or unwanted T cells that can lead to autoimmune diseases. Interesting. Okay, so that was the review. I wish that there was a little bit more editing of it before publishing, but it is what it is. It is what it is. So, thoughts. Well, I can see a thought. So with the Mhc multimer complexes, I can see those being very beneficial in the context of removing autoimmune diseases, especially. So we know for a fact that viruses, such as influenza and coronaviruses, they frequently will mimic proteins within the body, specifically peptides that are within the body. And this is one of the evasion techniques that viruses use to not get to avoid antibodies. Because if the antibodies are being produced against your own tissues, they end up being sequestered and not basically rendered useless. So one of the ways that we know viruses do that is by imitating the corticotropin, or ACTH, and this ends up creating antibodies against ACTH, and that ends up resulting in suppressed cortisol release downstream. My concern is that there's probably a lot of other diseases, or sorry, a lot of other peptides that these viruses are imitating that we just haven't had research to discover yet. And in fact, there's a plethora of autoimmune diseases that are the result of peptide hormones being sequestered by autoantibodies or self-attacking antibodies. So this is and there are a lot of diseases such as Alzheimer's, dementia, multiple sclerosis that are all implicated in these autoantibody attacks. So it is possible that if we use these major histocompatibility complex multimers for therapeutic use, it could be possible that we start eliminating these autoimmune responses. So that would be an area of research that, frankly, would be pretty good, especially in the context of these viruses being produced, these viruses that are spreading around all the time, screwing up our immune system. So we like to think that, you know, as our immune system becomes better and better as we get older, but what is really happening is our immune system is starting to make more and more antibodies for more things, and for every new antibody, there is more, what would the word be? There is a lot more opportunity for those antibodies to start attacking your own body. So keep this in mind. The more diseases that you get infected with, the more antibodies, the more variety of antibodies you are producing. And the more variety of antibodies that you are producing, that is another opportunity to have an antibody that can attack something in your own body. Okay, there's a limited number of protein sequences possible, right? So since there's a limited number, there's a lot of possible cross-reactivity or crossover that takes place. So what I'm imagining right now is that as people are aging, they are making a greater variety, or through their life they've created a larger variety of antibodies. And what I'm seeing is over time we actually do lose a lot of the antibody concentrations until we get infected again with that particular disease. And so in fact we actually will lose immunity to some diseases, right? But what we won't lose is the autoimmunity. We will maintain an autoimmune response. So if you start producing an antibody that was originally for a virus, but it starts attacking your own peptide hormones, or it starts attacking something in the body, it is those antibodies, or the cells that produce those antibodies, they are going to, it'll be much more difficult for them to be reduced over your lifetime, because those cells keep on getting activated by running into those proteins within your own body. So this is why autoimmune diseases are so freaking painful and complex and hard to deal with. So I'm imagining a scenario over time, and someone could actually run a simulation about this. Over time, you make more antibodies, and then you are keeping the antibodies that attack your own body, and over your lifetime, you are keeping the antibodies that attack your own body, and you are losing the antibodies that attack pathogens, because that's just how the system works. You can't be always producing high levels of every antibody that you've ever come across with, because that requires way too much energy. So eventually, after the disease has gone away, the antibody concentrations will go down and down until you get infected again with something similar enough to elicit that response. So I'm very easily imagining a situation or a scenario where, as people get older, their immune system goes more from attacking what is foreign and shifting more and more towards what is attacking your own body. Yeah, that is definitely a possible scenario, which could have large implications in aging, right? So as you get older, you're making more antibodies that just attack you, and it's hard to downregulate those antibodies because it's you. The immune cells are always being activated, especially if they're peptide hormones. And so if that is the case, if that is happening, then your body is going to start accumulating damage progressively faster, the older you get. I mean, that would be a model that someone could, run simulations on to see if that is actually plausible. There might be some sort of negative feedback loop or some control mechanism, or there definitely has to be some control mechanism in the body to help prevent that from happening. But yeah, from what I'm imagining is that over time, your immune system just becomes more and more caustic to yourself, because you're always being exposed to viruses, and these viruses are mutating, and these bacteria are mutating, and so over time, you're going to run into some pathogens that look just a little bit like you, at least have proteins that look a little bit like you. So this is one of the reasons why I was so concerned about the SARS-CoV-2 coronavirus. So the SARS-CoV-2 coronavirus did have a section, a full-fledged peptide, and you can look at one of my previous videos where I discussed it. So it had a sequence of 15 peptides, or 15 amino acids, that was near the beginning of the spike protein. And it had 9 amino acids that were identical and in the exact right location. So 9 out of 15, that's already very similar and very hard to accomplish. But not only did it have 9 out of the 15 identical, it also had 4 amino acids that were chemically very similar. So, amino acids, they have different residues on them, and the different residues will have their own chemical properties. So some amino acids are much more similar than others. So for instance, serine has an alcohol group on it. Threonine also has an alcohol group on it, and so frequently, if a serine and a threonine replace each other, usually there's similar chemical properties between those two. Okay. Same thing with aspartic acid and glutamic acid. They're both acidic residues. And so if one replaces the other, one of them's a little bit bigger, but it still has very similar chemical functions. So with this particular sequence, amino acid sequence, at the beginning of the SARS-CoV-2 spike protein, there was a sequence of 13, it was 14, 15 amino acids, and 9 out of those 15 were identical, and 4 were chemically very similar. So at some point, it was pretty much. I forgot to mention what it's similar to. It was very similar to a peptide hormone in the body called the NPP, which stands for, I believe, the N-terminal peptide of the pro-opiomelanocortin proprotein, so NPP. And this peptide is also implicated in the suppression of the release of prolactin. So if you end up making antibodies against this particular amino acid sequence, and you can look at my previous video to see what sequence it was, if you make an antibody against the sequence, so this would result from a dendritic cell or a macrophage or a B cell consumes a virus, consumes the spike proteins, and then the spike proteins get broken up by peptidases or proteases into smaller peptides. These smaller peptides get loaded onto the major histocompatibility complex class 2, and then they get presented on the surfaces of these cells. And then this can activate the T cells. And then the T cells will start making, I don't remember if it's the T cells, but basically your immune system starts making antibodies against these peptides. But this peptide looks very, 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 very similar to this NPP peptide. And so, if you make an antibody against the virus peptide, it can cross-react or can start auto-attacking your own body's peptides, your own body's NPP, and then also, as a result, from that interaction, the virus actually can survive a lot longer, it can duplicate a lot longer, and then oftentimes you have symptomology associated with the dysregulation of your immune system going into a pro-inflammatory state, causing symptomology. You become more leaky, you end up coughing, spraying, what have you. So my worry right now is that SARS-CoV-2 does have a sequence that is so incredibly similar to human hormone, and like mammalian hormone, because it's a hormone that is conserved among a lot of animals. My concern is that we will probably see serious autoimmune responses against that are caused by that peptide, okay, which could seriously affect prolactin levels for an extended period of time, which could probably cause just general hormone dysregulation, fatigue, basically mimicking the effects of pregnancy and the effects of lactation, right? So, yeah, I will look into it more. But there's a reason why I wanted to go over this review of the major histocompatibility complex, even though I skimmed it earlier and I screwed up with, because remember, these people aren't native English speakers, so there can be a little bit more difficulty in having them write things syntactically perfect. So sometimes it's hard to parse what exactly they're saying for a few sentences. But what I will say is that I want to go over this review just so that I can have a video log of it, and also that there can be just general education about how the immune system functions and how these receptors are utilized by the immune system in order to cause the creation of antibodies and cause the immune system to adapt to attacking a disease, and how that can get screwed up, how those wires can cross in a bad way to cause autoimmune diseases. So, and I also wanted to bring it up because we went over a paper, we went over a review on, what was it, testicular function and the immunoprivilege of the testicles, because that was, I believe, an important subject, but they brought up the major histocompatibility complex multiple times, so it's good to have a refresher on this entire process, so that everyone can be on the same page. So with that, I think I'm going to call it quits, and thanks for listening, and I will see you tomorrow. If you have any questions, leave them in the comments. Thank you.